Okay, good evening, Bio 202, or sorry, Bio 205 students. Welcome to our Unit 2 exam review. If you're in Dr. Cotter's Monday class, some of this will be very similar to what we went over today. If you are in Dr. Cotter's Wednesday class, welcome. I'm Luis. I am Dr. Cotter's Monday Success Specialist. Um, Vlad is sick right now, and so I'll be handling the exam review for him this week as well. If you need to contact me, just reach out to Dr. Cotter, or I know I've got like half of the students in there that are in my 202 class, and they know how to get a hold of me. So I'll go ahead and kind of go over the Bio 205 review, talk about some of the things that we need to look out for, key pages, and then I'll go over some practice case studies or practice cases that Dr. Cotter prepared for us. Okay, so we did a lot of stuff in this unit, kind of going back and thinking about why we did it. Basically, the whole purpose of this was how do we kill microbes? How do we disinfect? How do we sterilize? How do we keep ourselves safe from microbes? That's majority of this unit. So the first parts are ways to describe or to organize microbes, by either their oxygen requirements or if they like salt or not, things like that. And then once we understand the structure and the environments that microbes like to live in, we can then tailor solutions to kill or negate or neutralize these infectious agents. So really quickly, if you have one drop of hand sanitizer, it'll kill 99.9% of germs. Obviously, if you do two drops, you'll kill over 100% of the germs. Going through this unit, you should understand that's not actually what happens, um, but just an understanding of, hey, we sterilize, certain things disinfect, and things like hand sanitizer are antiseptic, which are for living surfaces. So the agenda will go over nutrient, nutrition, and growth for microbes, genetic and reproduction, methods of control, antibiotics. I'll go over a practice case, and I'll show you how to access those cases, and then I'll answer any questions. Okay, the first one, Monday students, you should have already seen this quiz go up online. Wednesday students, you may or may not have this quiz yet. It might not become available till till Wednesday. But essentially, for the exam, that's going to be held next week, either Monday or Wednesday, based on your class, will be done preferably on your own devices, either laptops or iPads. We will have the devices available in the lab if you need to borrow one. But the reason we're doing your device is that if you looked at the syllabus, you'll realize that Thanksgiving the week of Thanksgiving is falls on the same week as your Unit 3 exam. We're kind of talking about potentially having that exam be done remotely. So this is going to give us an opportunity to see if your device will be able to use Respondus. That way it's ready in case we decide to take the exam. Online, virtually, from wherever you're at, either Monday or Wednesday, the week of Thanksgiving. So please bring your device. Obviously, if you don't have one or if you can't bring one, we still have devices available so that you can take your exam, but preferably you bring your device so we can kind of test whether it'll work for a Unit 3 exam. And that's why we're asking you to bring them this time around. Biggest thing, in this link, you'll see that there's specifically when you go download Respondus, if you are in multiple colleges, I know like when I was in ASU, we have our own different Respondus link uh, compared to Maricopa Community Colleges. So if you haven't tried Respondus at any class here in Maricopa Community Colleges, you want to download the Maricopa specific Respondus. That just lets the Respondus team know that that license is for Maricopa Community Colleges, and that'll let you open our exams versus like an ASU browser. Okay, so I'm going to kind of very quickly go over these next few ones because they're very minor in terms of the exam. You might get like a few questions, but it's not going to be a majority of your exam. So growth phases. So remember that we usually start in 4 different phases. You have your lag phase, your log phase, your stationary phase, and then your death or decline phase. So the lag phase is basically your microbe um, may not have just have been just introduced into your body. So it is starting to multiply, but it's not multiplying all that quickly. One cell to two cells, two to four, four to eight, eight to 16, etc. It's a very slow start to the process. The log phase is where you start getting an exponential growth, where the bacterial cells are now able to multiply so quickly that the quote unquote infection really starts. We'll kind of talk about the phases of infection in unit 3 or unit 4. But essentially, what happens is that you have a buildup of these microbes in your body, and then your immune system will try to fight those off. And then, depending on how quickly our immune system can fight off the infection, will determine how deadly or severe or harmful that infection could be. In our case, the bacteria, specifically bacteria, they have a big log phase where they're able to rapidly consume nutrients and resources, and they're able to grow really quickly. At a certain point, there's not going to be enough resources for them to grow quickly, so they become very stagnant. There's just enough food to keep the existing bacteria alive, but not enough food for them to then multiply and reproduce. And then after a while, these bacterial cells will start running out of resources, and then they'll start the decline phase or the death phase. Again, that's more detailed than that's actually needed. Biggest thing to understand, the lag phase is a very slow increase in microbial cells. The log phase is an exponential or rapid increase. Stationary is a steady, where, for every one cell that dies, another cell replaces it. And then the decrease phase, the death phase, where those microbes start to die off because there's no longer any resources to keep them alive. Environmental conditions. Okay, this one we kind of, for the Monday group, we did this quiz um, with Dr. Cotter, and this was one of the ones that we really struggled with. And that's okay. We haven't seen this in probably almost a month, if not more. So just to kind of throw these out there to kind of reintroduce this so that you're ready for the exam. So the temperature ones, you guys did pretty well with this one. So remember that the psychrophiles they like the -5 to 20 degree conditions. Mesophiles, 20 to 40. That's primarily what we worry about as human beings. Thermophiles, 40 to 80 degrees Celsius, and hyperthermophiles, anything greater than 80 degrees. The oxygen requirements, like I mentioned, this is what we started with in class. Aligned here, you have your strict or your aerobes. They're called obligate aerobes, but just remember aerobes for this class. You have your strict anaerobes, or we'll just say the anaerobes. Again, they like growth away from oxygen, whereas the aerobes like growth closest to the oxygen. Then you have your facultative, which this picture doesn't really do it justice. Just remember that facultative grows all along the tube, up and down, and everything in the middle. Aerotolerant aerobes, they are aerotolerant microbes. Uh, they can tolerate oxygen, but they don't really like it. So you'll have more of a growth like in the lower half portion with occasional like growth spread across the tube, but it's not going to be a very heavy tube like the facultative anaerobes would have, or the facultative microbes have. And then microaerophiles, those grow not on the surface, but right beneath the surface. They like that 5 to 10% of oxygen. So take a look at your notes, especially because those pictures are more related to the ones you'll see on the exam. But like I said, we definitely were about 50-50 on some of these questions when we took our practice quiz today. So just study these. That way, these should be pretty straightforward for y'all. And that way you don't lose some easy points. Okay, next we have pHs. The way the pH question is going to be worded is you might be asked, hey, this pH or this microbe likes a pH of less than 7 or an acidic condition. So you'll know that acidic or acidophiles like a pH less than 7. Neutral or neutrophiles, they like a pH of 7. give or take, 6.5 to 7.5, something, things like that. And your basic or your alkalinophile likes the pH greater than 7. Um, More likely than not, for the way the case is going to work is you might be given a pH condition like this, and then like we saw in the practice quiz today on Monday, and for the Wednesday group, you'll see it on Wednesday. You want to kind of know where in the body those pH conditions exist. So acidic, that is your stomach. Primarily, your stomach is one of the most acidic places in the body. Also, the reproductive system, external openings. So the vagina, for example, that is also acidic. Um, Basic or alkalinophile, primarily that's your intestines, your small and large intestines. Those are primarily a above 7 pH. And then the neutral or neurophiles, pH of 7, typically your blood is like, for the purposes of this class, blood is neutral. It's like slightly acidic, but like not by much. So we'll just say blood is neutral. So that's where your neurophiles would be residing. So just for your exam, if you get a question about where would this acidophile live in the body, you'd say things like the stomach or things like the reproductive system. But if it's a pH of greater than 7 or an alkalinophile, you'd say, oh, that would be in the intestine, the large intestine, the small intestine. And then neutrophiles would primarily like in an environment that is blood heavy. So things like your blood vessels or blood is where these neutrophiles would typically try to reside in our body. And then salts, halophiles and halotolerance, just different salt conditions will attract different kinds of microbes. This one's not, honestly, too, not that important. The pH ones for sure, and then definitely the oxygen ones will be tested. Products of fermentation. This class has actually done very well with this one. I've had a lot of classes before that still miss this one on both the exam and the case studies. I think y'all have done really good with it, but just to refresh, fermentation will lead to three different things. It'll lead to alcohol, the beers, the wines, etc., mostly wines. It'll lead to CO2 gas, and it'll lead to acid. So it'll lead to one of these three different things, or sometimes they're a combination of the two, or a combination. Again, y'all have all done very well with these three. I haven't seen you miss these questions on the case studies. But on the exam, just remember these three because I have seen this as one of those things that people forget. It seems pretty straightforward, and the exam asks you, and then people just kind of freak out and panic. They kind of forget. Water movement. You have different things. So water movement cell-wise is how much water is in a cell versus out of the cell. And this is controlled primarily by sodium or salt. And so water likes to follow salt. So if you are a cell that's isotonic, you are very well balanced with sodium inside the cells versus sodium outside the cells. So because you're balanced sodium-wise, water will not move out or into a cell. Hypotonic solutions, you have more sodium or more salt inside a cell. And so what happens is that the water will then go follow the sodium inside the cell, go through the membranes, and eventually the cell will swell up to the point where it'll lyse or it'll burst open. And then hypertonic, you have more salt outside of the cell. So the water will leave the cell to go to the outside regions to follow the salt, and the cell will typically shrink. And then after a while, the cell won't actually be able to function because it's not, it's one, too small, and two, it doesn't have enough water to actually let everything kind of move around. Again, not a very big important question. Biggest thing is understanding where the salt is and understanding that water will follow that salt. Okay, bacterial genetics. So we've talked about three different types of things for bacterial genetics. We call this horizontal gene transfer, which means that the bacteria aren't transferring genetic information by reproducing like we would if we had children. They are just giving their genes to other existing bacteria that are not related or they're not, part of the same lineage, if you will. This can be divided three different things. You have conjugation, which involves the sex pilus. The plasmid will get copied over and transferred into the new cell or the recipient cell, and then that new cell will gain antibiotic resistance. It'll gain the ability to develop a sex pilus, and then it could then go and infect other cells and transmit that plasmid. Remember that plasmids are an extra piece of DNA that's not necessary for survival, but does give these bacterial cells the ability to get antibiotic resistance, um, develop the sex pilus, and then give it some other features. Transformation is where a cell is able to take in uh, information from other cells around it to then become, let's call it a super bacteria, if you will. Things like E. coli are what we call competent bacteria, where they will actually hunt and kill other bacteria and steal their genetic information. We also did this transformation in the lab. If you recall, we took our cells, we heat shocked them. In our case, you can also electrically shock them. And then we added the plasmid to our little vial with the bacterial cells. And some of our cells took in that plasmid and then developed antibiotic resistance. And then some were also able to glow. This is also a easy way for us as people to make antibiotics. This is how I made it in the lab. Is I'm able to take a plasmid with an antibiotic, give it to E. coli. Coli is then going to reproduce every 20 minutes. And over the course of a week, I've taken my one little plasmid of an antibiotic, and the E. coli has made hundreds of thousands of that strain of antibiotic. And then I can go and kill the cells and take the antibiotic and then ship it off to whatever. We used to do this at ASU for our experiments. But again, just transformation is you're giving something that's living an extra piece of information, either from its environment, and then it's able to develop those extra traits that you give it into its normal cell. And then transduction, this is what we're working on in the Monday group, where we have infected our bacteria with phage or bacteriophage, and the bacteriophage DNA is accidentally replaced by another cell's DNA or another bacteria's DNA, and that gets transmitted to a separate bacteria that then gains that DNA fragment of the bacteria that the phage or the virus stole it from. Okay, energetic drift versus energetic shift. Biggest things for this one, the key factors. Drift is a small or a minor change to a viral spike protein. It's a slow change, and it leads to what we call point mutations. And energetic shift is a major change in viral spike structure. It is very quick, and it is caused by or could lead to things like genetic recombination, where we create novel or brand new viruses because of the fast change. These are really the big ones, the big keywords to look at when you're doing the exam. Don't worry about too much about the specifics of it. Just know that the difference between drift and shift. Okay, so this is where we actually get to the major part of the exam. It's going to be all about methods of control and then antibiotics. So that's going to be page 7 and page 10 of your study guide. So I broke it down. I organized some of these methods of control by physical on the left and chemical on the right. You can see I organized it by things that sterilize. So physical, things like the autoclave at 121 degrees Celsius at 15 psi, ultraviolet C at 260 nanometers, and things like filtration, which will sterilize but will not kill. And then on the chemical side, we've got our O's, the aldehydes, the halogens, and the hydrogen peroxide. Specifically for sterilization, hydrogen or halogens and hydrogen want to be in a gas form. That's the best way to sterilize. Whereas disinfectants can be more liquids like your rubbing alcohol, your isopropyl alcohol that you have, or your hydrogen peroxide you have here at home. And then some methods of disinfection, things like boiling, pasteurization, dry heat. For physical, chemical, we have 10% bleach, 70% ethanol, and then pine sol Lysol. There's just like a quick way of organizing it if you're still trying to find a way to organize it. The list that you want to know is the list on page 7 that has all of these physical and chemical methods, and whether they sterilize, they disinfect, or they're antiseptic. When we get to the practice cases, I'll just show you all how you can organize this to kind of help you prepare for the exam. And then antibiotics, page 10. Biggest thing about this one is know your drugs and know what they target. Dr. Cotter mentioned making things like flashcards to say that, oh, this drug is either goes against the peptidoglycan cell wall, the waxy cell wall, plasma membrane, etc. Um, for plasma membrane, this one is pretty easy because we only have one, and that's the polymyxin B. So if you see anything that targets plasma membrane, that is gram-negative plasma membranes, and you want to use polymyxin B. For the peptidoglycan cell wall, basically anything that ends in cillin, so penicillin, methicillin, ampicillin, amoxicillin, those all target the peptidoglycan cell wall. Anytime you see the word waxy cell wall, that's typically referring to things like tuberculosis. And we have ambunol and isoniazid. Those are really the only two that we use against tuberculosis. And then obviously you have the other ones, the folic acid metabolism, knowing which one is more or less toxic. And then protein synthesis drug antibiotics, also knowing which one is more or less toxic. And that should be on your page 10 of your study guide. And then looking at which ones target DNA, and then which ones are the ones that go against the viruses, the Tamiflu, Relenza, AZT, that kind of thing. Okay, so what I'm going to share with you all after this in the announcement is this practice cases for BIO202. So it's going to take you to this link here. Mine is an editable version. When you get to it, it's going to be view only. So what I want you to do is go to File, make a copy, and then this will make a copy on your Google Drive that you can then edit. But the way these are grouped is that Dr. Cotter gave me 3 cases that I can share with the group. So you have one that's Mycobacterium tuberculosis, hint, hint. We just talked about tuberculosis. Another one is an endospore forming bacteria in the GI tract. Biggest thing to note when we talk about the cases on not just Unit 2, but Unit 3 is what type of microbe is it and where in the body is it located. And then group 3, Pseudomonas aeruginosa in a water system like air conditioning or your HVAC for the hospital. So when you work these cases, basically, this is like this is going to look very similar to your case studies on the exam, where you're going to be given an example, like, hey, Mycobacterium tuberculosis in a surgical room, and you're going to want to say, okay, what type of microbe is this? So tuberculosis is an acid-fast bacteria with a waxy cell wall. And I gave you all this already, so you don't have to go find it yourself. Then your next questions that come off of the exam, and these are probably going to be typed questions. Just, if you're listening to this, just prepare for that. You're going to be asked, hey, give me 2 methods to physically decontaminate the non-living surfaces. So you'll go through, all right, I'll write my 2 methods. Then it's going to be like, hey, give me a method or 2 methods to sterilize these microbes. And you kind of see that we gave you the definition here. Again, what you're going to go down here is you're going to type in things like, oh, I can autoclave, or I can do UVC, something like that. Feel free to type on this because it's more for you to kind of practice writing this stuff down. The more answers you put on here, the better, because you can then study this. And when the exam happens, we're not going to ask you for all of them. We're going to ask you for 1 example, 2 examples, 3 examples, that kind of thing. So you go down to the physical methods to disinfect. You'll type some of those out. You're gonna be like, all right, what chemical methods decontaminate or sterilize? Okay, I know I have my Oz. So you'll write down your Oz. What disinfectants do you have? And then what is very good for antiseptics? Primarily pay attention to things that say residual. and non-residual. So you might be asked for a question, hey, give me a residual chemical that will stay on the hands, and then give me a non-residual, something like an alcohol that evaporates very quickly. And then you'll have a question towards the end. Hey, what drugs can I give my patient for this tuberculosis acid fast bacteria? And kind of like I mentioned earlier, is we have two drugs. It might ask you for one or the other. And I'll do oh God, ethambutol and isoniazid, for example. And these are really the only two that you should be putting on here. So as long as you remember one or the other, you should be okay. For the ones that we have labeled on the notes for more toxic, less toxic, if we're giving you a patient to treat, you probably don't want to give them the more toxic drugs, give them the less toxic version. So things like trimethoprim, which is used for metabolism, that's usually safer. Or things like erythromycin for protein synthesis is safer than things like tetracycline or chloramphenicol. Um, but again, that's all as you organize this here, put that in there so you can start doing it. Also, things like what do I say? Flashcards are very good for connecting drugs to their cell targets, things like that. And then just kind of go through, endospore forming. I guarantee that the endosperm question is going to ask you the three layers of protection. So I already gave them to you here, but just study those, just remember them. They haven't changed since Unit 1, but I've seen students miss easy points on this because literally the first question is, what makes this thing so much difficult to kill? Oh, it's because it's got the three layers. What are the three layers? Calcium, keratin, peptidoglycan. That's it. So don't miss this one because again, it's fairly easy points. Just students typically tend to either not study this or not go back to this because it seems so simple. But it's always when you get to the exam, you see the simple question and you just like blank. It's happened to me. It's happened to many students. So try not to do that here. And again, you'll just go through, all right, this is an endospore forming. It's in the GI tract. I'll write some physical methods to sterilize, to disinfect, chemicals, drugs. Um, and slash antibiotics. And then one more, which is a Sumo aeruginosa is a gram negative in a water system. And then you kind of, this forms this like waxy or this sticky layer here. And again, physical, chemical methods, antiseptics, drugs, etc. The format of these practice cases is going to be near identical to the exam. So do all of these, write in, put in the answers, try to, solve these for yourselves. Once you're done, if you would like for me to review these, just send me the link. So you can just do share and then type in, Luis Salcedo or Luis Salcedo at phoenixcollege.edu. Let me know you want me to take a look at it, and then I'll go and come in here and I can like highlight things and, leave comments like correct or something like that. Or I'll tell you, hey, take a look at this page or add some more detail here, that kind of thing. That way you know that your practice cases are good enough to actually study off of it when you get to the exam. Okay, that's really all I have. Trust yourself. Don't second guess yourself. I believe in you. I know you guys can do it. Happy Halloween. With that being said, does anyone have any questions? You can either unmute or type it in the chat. Okay, if you have no questions, then again, thank you very much for joining me this late. I appreciate that you made the time. For those that were not able to join, enjoy the video recording. If you have any questions, please reach out. Otherwise, have a good night, and I will see my Monday group on Monday, and then Wednesday group. Hopefully, Vlad's doing okay, but if you have a second, please reach out to him. Just let him know that you're thinking about him. Wish him well. Wish him hope he gets better, so he can join you all either Wednesday or maybe next week. All right, I'll talk to you all later.